So welcome again guys, welcome back to my channel, and for today we'll be discussing, as you know, we have completed our first kingdom, that is kingdom Monera, in my previous lecture. So before starting the new kingdom, I think we have to solve some questions, right? So NEETATHON. So the first question is, the peptidoglycan cortex of bacillus spore have. So if you remember, we have discussed about the structure of the bacillus spore, right, which was one of the asexual reproduction in bacteria. So in that we have discussed that the cortex layer of this spore has an anticoagulant as well as a dipicolenic acid and calcium ions. So we have both of these. So option C will be correct. Now moving towards the next question is, consider the following regarding the reasons for the fact that now cyanobacteria are kept in Monera and not in Plantae. So we have to find a way, or we have to find those features or properties or the points जो कि हमें ये बता रहे हैं कि why they should kept in Monera only, not in Plantae. So the first point is they are prokaryotes. Right, so yes, they are prokaryotes, that's why they are kept in Monera. The second point is the cell wall of cyanobacteria has peptidoglycan. Yes, that is also an exclusive feature of Monera, which means they have a cell wall of peptidoglycan. And third one is they can fix atmospheric nitrogen. So no, this can't be a defining feature for our organism to be kept under kingdom Monera, because it can be there are many nitrogen fixing bacteria, but nitrogen fixation property cannot be a feature, or it is not a necessity for an organism to be included in kingdom Monera. So we have the first and second options correct. So the correct option will be the first one, wherein it is written first and second only. The heading towards the next question then. So identify the group that is not matched to all the characters shown. So the first is Monera group, cell type prokaryotic, ठीक है, cell wall absent, no, ये गलत हो गया यहाँ पे. सेल वॉल एब्सेंट नहीं होती है, सेल वॉल तो बल्कि पेप्टाइडोग्लाइकन की बनी हुई होती है. सो यहाँ पर ये गलत हो गया. देन द न्यूक्लियर मेम्ब्रेन एब्सेंट, राइट. द बॉडी ऑर्गेनाइजेशन सेलुलर, राइट. सो द फर्स्ट ऑप्शन इज रॉन्ग ओनली, राइट. तो यहाँ पर ऑप्शन कौन सा रॉन्ग हो गया? द फर्स्ट वन ओनली. बाकी प्रोटिस्टा यूकैरियोटिक, प्रेजेंट इन सम सेल वॉल, न्यूक्लियर मेम्ब्रेन प्रेजेंट, बॉडी ऑर्गेनाइजेशन सेलुलर, करेक्ट. Fungi, cell type eukaryotic, cell wall present, nuclear membrane present, body organization multicellular or loose tissues, correct. Plantae, eukaryotic, cell wall present, nuclear membrane present, body organization tissue or organ. So all three are correct. Only the one option which is not matched correctly is the first option, that is Monera. Now heading towards the next, identify the incorrect statement regarding bacteria. Bacteria are the most abundant microorganisms. Okay. The bacteria also live in extreme habitats where very few other life forms cannot survive. Yes, we have thermoacidophiles, we have halophiles, we have methanogens. So correct. Bacteria as a group show the most extensive metabolic diversity. That means these the first, second, third, three options are correct. Now the fourth option is they are obligate intracellular parasites. This is not correct. This is incorrect. So the incorrect option will be the fourth one, है ना? Because ये obligate intracellular parasites नहीं है. Obligate means we have two words. The first one is obligate and the second one is facultative. Facultative. So you have studied about obligate aerobes, obligate anaerobes, facultative aerobes, facultative anaerobes. So obligate means they can't live without that particular thing. For example, obligate anaerobes means they are obligate anaerobes at any conditions. But facultative anaerobes means they are anaerobes but they can survive even in the presence of oxygen too. So मतलब they are not strictly regulated, ठीक है ना. बट यहाँ पर क्या लिखा है? ऑब्लिगेट मीन्स दे हैव टू बी द इंट्रासेलुलर पैरासाइट्स ओनली. Extracellularly they cannot live, but that is not there. That is not the concept, or that is not the point here in bacteria. So this one is incorrect. The next question is, identify a character that is not unique to Archaea bacteria. So the first one is cell wall structure, the second one is cell membrane structure, the third one is flagellin protein structure, and the fourth one is fatty acid synthetase present. So if I have to identify which is not unique, so cell wall structure is unique to Archaebacteria. It has lipopolysaccharides. Then cell membrane structure is also unique. Flagellin protein structure, okay. But fatty acid synthetase present, this is not unique. ठीक है ना? So it is not unique to Archaebacteria. Rest three are unique to Archaebacteria, right? So the next question. So the next question is, Red Sea was named due to which reason? So the first option is excessive growth of blue-green algae. This is correct. The blue-green algae's name was Trichodesmium erythraeum. The second option is red pigment by fishes. This is wrong. The third option is abundant growth of Trichodesmium. Yes, this is right. And D is both A and C. So yes, option D will be correct, right? Then heading towards the next question is, consider the following regarding mycoplasma. So the first is, they are genus of bacteria that have a unique cell wall of their own. So is it correct or not? So mycoplasma are the smallest organisms and they don't possess cell wall. That is the reason they are known as joker of plant kingdom, because they are pleomorphic in nature. So this stands incorrect. In their genetic code, UGA codes for tryptophan. You know, UGA is one of the stop codons, so it don't code for anything. Now none of them is pathogenic to humans. Yes, it is pathogenic. We have seen the urethritis occurring in humans. Then they have a large genome. So they have a large genome. No, their genome is very small. Fifth one is, they can be killed by penicillins. So penicillin only affect those organisms in which there is a cell wall, but these organisms lack cell wall. So they cannot be killed by penicillins. They remain unaffected by the presence of penicillins. So this is also wrong. So the last statement is, they can survive without oxygen. Yes, this is correct. So the number of correct statement is only one statement. So going into the next question is, what are the features of Heterocyst of Cyanobacteria? So it has both PS1 and PS2. No. It only has PS1. Yes. Its wall is impermeable to gaseous exchange. Yes. So that means the both B and C. So you remember the Heterocyst, all the other vegetative cells of Cyanobacteria was having both PS1 and PS2. But only the Heterocyst had only PS1, and this was due to the presence of nitrogenase enzyme in the heterocyst, and this nitrogenase can get oxidized by photolysis of water occurring in PS2. So in order to prevent the oxidation of nitrogenase enzyme and like उसको वो unfunctional बना दे, so उससे अच्छा क्या हुआ कि उसने एक adaptation की and heterocyst ने पूरी तरीके से PS2 को eliminate कर दिया. So the next question is, the main difference in gram positive and gram negative bacteria resides in there. The first option is cell wall. The second option is cell membrane. The third option is cytoplasm. And the fourth option is flagella. So that we have studied the cell wall structure of both the gram positive and gram negative bacteria. So yes, definitely option 1 is correct, cell wall, because the rest of the properties do not varies considerably. So the main difference in both of these bacteria resides in their cell wall. As you know, I have told you that gram negative has 2 layered cell wall, whereas gram positive has 1 layered cell wall. Also the gram negative 2 layered cell wall is made up of peptidoglycan and muramic acid and lipopolysaccharides and lipoproteins, whereas plus or positive strain bacteria, gram positive bacteria, is made up of peptidoglycan and teichoic acid. So that we have studied in our lecture. So the option will be cell wall. So the next question is, name the organisms which do not derive energy directly or indirectly from the sun. Those organisms which derive this energy directly from the sunlight will be known as photosynthetic bacteria. So chemosynthetic bacteria will not be using sunlight in any of the form, and that's why the answer of this question is chemosynthetic bacteria. So pathogenic bacteria can also derive indirectly, symbiotic bacteria can also derive indirectly, mold can also derive indirectly, but chemosynthetic bacteria do not derive this energy directly or indirectly from sun. So the option remains option A. Then the next question is E. coli or Escherichia coli is used extensively in biological research as it is. So the first option is easily cultured, easily available, easy to handle and easily multiplied in host. So as you know कि there can be many reasons है ना, but the main reasons behind E. coli being the model bacteria or the main organism to be like experimented upon is that it is very easy to handle. ठीक है, so it can be easily handled in a laboratory. So that is the main reason behind biological research use. The next question is genophore or bacterial genome or nucleoid is made up of A. histones and non-histones. No, because the DNA of bacteria is naked. So what do you mean by naked bacteria? So naked bacteria means that you don't have histone proteins. So the first option remains incorrect. The second one is RNA and histones. Histones again no, can't be there. So first incorrect, second incorrect. Single double stranded DNA or a single stranded DNA. So if you remember this was the I guess mesosome, and then from here you can see the nucleoid attached which is double stranded, right? If you have seen the structure properly then you could have observed this. So the double stranded DNA is present and a single double stranded DNA is present, so the option will be C. The next question is transduction in bacteria is mediated by. So the first option is plasmid vectors. No. Phage vectors. Yes. You remember that we always see bacteriophages. What are bacteriophages? So bacteriophages are those viruses which infect bacteria. So the phage vectors is the answer. The next question is which type of DNA is found in bacteria? So circular free DNA, membrane bound DNA, straight DNA or helical DNA. So you have seen that it's a circular free DNA present inside the bacteria, right? So the next question is the site of respiration in bacteria is ribosome, microsome, episome and mesosome. So the site of respiration we have discussed in which part? In mesosomal part. So mesosome initiates or it helps in replication as well as respiration. Okay, so the answer will be mesosome. So the next question is a few organisms are known to grow and multiply at temperatures of 100 to 105 degrees Celsius. They belong to. So temperature means something has to be related with the word thermo, है ना? So यहाँ पर क्या है? Thermophilic sulfur bacteria. So this should be the answer. What do you mean by philic? It means thermal or temperature loving species. So hot spring blue green algae no, methanogenic no, marine no. So it is thermophilic sulfur bacteria. The next question is a bacterium divides every 35 minutes. Okay, so its dividing time is 35 minutes. If a culture containing 10 to the power 5 cells per ml is grown for 175 minutes, what will be the cell concentration per ml after 175 minutes? So first of all you have to calculate how many cycles it has undergone. Okay, so we can see that 175 are the total minutes or the time, and a single cycle takes 35 minutes. So if we divide it, it can be 35 5s are 175. So it means we have 5 division cycles. Now as you know, initially the culture was containing 10 to the power 5 cells per ml. So after 5 cycles, what will be the amount of cells present? So for that we have to apply a formula known as 2 to the power n, right? So if we calculate 2 to the power 5, then we will get 32. Now so after 175 minutes your cells will become 32. and you have to multiply it with the number of cells which were initially present. ठीक है, so 32 into 10 to the power 5 in total cells will be present. So the option B will be correct. Now heading towards the next question. Transfer of genetic information from one bacterium to another in the transduction process is through. So we have discussed it earlier to the bacteriophages, right? So the bacteriophages released from the donor bacterial strain will infect the other bacteria, and the transduction and the transfer of genetic information will take place from one bacterium to another, and the process will be known as transduction. The next thing or the next question is, photosynthetic bacteria have pigments in. So the bacterial pigments resides in chromatophores. That you need to remember, we have discussed also. So chromatophores bears all the pigments considered with the bacterial part. The next question is, what is true for cyanobacteria? So it is oxygenic with nitrogenase, oxygenic without nitrogenase. So first thing you have to see, it is oxygenic or it is bearing or not bearing nitrogenase, right? So as I have told you earlier that it is the only bacteria which is oxygenic in nature. So it is oxygenic. So first part is clear. The second part is, it bears nitrogenase or not. So ये भी अभी हमने बात की in the previous question कि due to the presence of nitrogenase in heterocyst, the cyanobacteria lack PS2 in heterocyst cells. That means they bear nitrogenase. So the option will be the first option, that is oxygenic with nitrogenase. Now the next question is, which one of the following statements about mycoplasma is wrong? So they are pleomorphic. Yes, this is not wrong. This is correct. They are sensitive to penicillin. No, they are not, because the cell wall is not present and penicillin work on cell wall only. They cause diseases in plants. So this is also correct. They are also called PPLO. Yes, this is also correct. So the main one of the statement is correct, that is the B statement. So the answer will be B. Now the next question is, an example of symbiotic bacteria. So Xanthomonas, Rhizobium, Erwinia or Agrobacterium. So that you know, Rhizobium is an example of symbiotic nitrogen fixing bacteria, है ना? So ये हमने पढ़ा भी है. And Rhizobium जो है, it is present in the root nodules of legumes. So legumes के root nodules में ये present होता है Rhizobium. The next question is, N-acetyl muramic acid is present in. So N-acetyl muramic acid is present in cell wall component of plant? No. The cell wall component of gram-positive bacteria? Yes. Cell wall component of fungi? No. Viral coat material? No. So the option will B will be correct. Now the next question is, a common character found in cyanobacteria and eubacteria is. So blue-green algae and eubacteria में क्या similarity है? So cell wall being composed of peptidoglycan? Yes. Occurrence of oxygenic photosynthesis? No. Cyanobacteria में oxygenic होता है but eubacteria में नहीं होता है. Presence of photosynthetic lamella? No. It is there in cyanobacteria but not in eubacteria. Presence of gas vacuoles? It is there in cyanobacteria. So this is also incorrect. So the first option is more correct, which is cell wall being composed of peptidoglycan. This is the common feature in both of these bacterial forms. The next question is, the select the odd one out regarding the basis of classification by R.H. Whitaker. So you remember that we have discussed around 5 bases of classification of R.H. Whitaker's system of classification, है ना? So those were mode of nutrition, those were the reproduction, type of reproduction, and the cell structure, the phylogenetic relationship, right? So which of the following is not there in the basis of classification? So cell structure was there, the mode of nutrition can be there, that was there, phylogenetic was there, but cell wall structure was not there, right? So the answer will be cell wall structure. The next question is Whitaker kept Chlorella and Chlamydomonas in which kingdom and later on kept in which kingdom? So Chlorella and Chlamydomonas was initially kept at kingdom Plantae, and then later on by modern taxonomy they were transferred into the kingdom Protista. So the answer is A only. The next is bacteria whose tuft of flagella present at one end called. So we have studied the types of bacteria on the basis of the flagella. So on the basis of flagella there can be many types. There were Atrichus means that no flagella is there. So that means no flagella. Lophotrichus means tuft of flagella at one end. So this gives the answer. Amphitrichus means the one flagella is present at both ends. So Lopho is one tuft, but Amphi means only single flagella present at both ends. Now Cephalotrichus means a tuft of flagella, two tufts. Matlab tuft of flagella are present in both the sides, right? And at last Peritrichus was when the flagella is present all over the body surface. So the answer to this question is B, that is Lophotrichus. The next question is purple sulphur bacteria is. So the purple sulphur bacteria is Thiospirillum. So we have studied about purple sulphur bacteria, purple non-sulphur bacteria, green sulphur bacteria, green non-sulphur bacteria, है ना? So purple sulphur bacteria was Thiospirillum. Rhodopseudomonas was purple non-sulphur bacteria. Chlorobium was green sulphur bacteria. Chloronema was green non-sulphur bacteria. ठीक है? So purple sulphur bacteria was Thiospirillum. The next question is a culture medium containing 1 into 10 to the power 18 bacteria grown for 210 minutes, which divides once every 35 minutes. So 210 by 35, we can calculate the total number of divisions. So when we calculate this, then we will get the answer as here 6, right? So the total number of cells will be 2 to the power n, that is 2 to the power 6, and that will be 64. ठीक है? So the total number of cells will be, initially there were 1 into 10 to the power 18 bacteria, now there is 64 into 10 to the power 18 bacteria. So the answer will be the option B. Now next question is a bacterial cell divide once every 2 minutes and took 61 minutes to fill a cup. At what time the cup will be 1/4 filled? So once after every 2 minutes it is dividing. That means it is doubling. So if you see a container which is filled with the bacteria and it took 61 minutes complete to fill a cup, ठीक है? सो अगर मैं 2 मिनट पहले देखूँ तो मेरी कॉन्सेंट्रेशन हाफ होगी. सो 2 मिनट पहले मेरी कॉन्सेंट्रेशन हाफ होगी. दैट मीन्स एट 59 minutes my concentration would be half a cup. Similarly, if I decrease 2 more minutes, that is around 57 minutes, my concentration will be 1/4. So यहाँ पर क्या पूछ रहे हैं? 1/4. So 1/4 के लिए आंसर क्या होगा? 61 minus 2 is 59. 59 minus 2 is 57. So 57 minutes पे cup will be 1/4 filled, right? So we have completed about 30 questions for today, which was very important and for Kingdom Monera. If you are able to solve these questions, so that means you are going great. And so keep practicing the questions. That is the most important thing right now. And if you have any doubt in any question or you have any suggestion, please drop it in a comment box, right? Also, if you like my videos, please like, share and subscribe. So that's all for today. Soon we will be coming with a new video with new kingdom, and we'll be starting with our new topic. So stay tuned.